right, so without further ado, let's get started. We're so excited to have our two esteemed presenters here this evening. Dr. Oswald Schmitz literally has a CV that's 31 pages long, and it's full and really interesting. He's currently at Yale as the Osler Professor of Population and Community Ecology, head of the Schmitz Lab there, and a Senior Associate Dean for Research and Director of Doctoral Studies in their School of the Environment. And notable to those of us in Ann Arbor, he received his doctorate from the School of Natural Resources here at the University of Michigan. His research helps explain how predator and herbivore species determine the species composition and productivity of plants and ecosystems, and his scientific insights help in the drive to conserve the essential services that species within ecosystems provide to humankind. His research evaluates how to rethink conservation strategies, offering alternatives to traditionally extensive and expensive approaches in environmental management. His newest book, and he has several, is called The New Ecology: Rethinking a Science for the Anthropocene, and is all about his studies on the linkage between biodiversity and ecosystems. We are also pleased to have Dr. Eric Lundgren with us this evening. Dr. Lundgren received his master's degree at Arizona State University and PhD from the University of Technology in Sydney. His research primarily focuses on ecosystems which have both native and introduced species, giving him a really important understanding of biodiversity and helping us all answer questions about not just what's happening now, but what's happened historically, even as far back as the Ice Age. And he's had a front seat into seeing different values that shape our science, which, while essential to guiding our responses to things like climate change, can also blind us in other ways. When Dr. Lundgren isn't studying interesting things like how donkeys dig wells that benefit other species, he's writing or presenting, but not with just peer-reviewed research, but also articles in Science, Smithsonian, Conservation Biology, and oodles of others. And I should mention he's been featured on Science Friday. I'm very jealous. Welcome to Dr. Lundgren. But first, our CEO and President, Tanya Hilgendorf, has a few words to share.
Thank you, Wendy, and thank you everyone for being here tonight, and to our esteemed presenters. We are grateful for your valuable time and attention. We all know wildlife in Ann Arbor are a well-loved part of our community and our surrounding communities. Deer specifically are admired for their grace and their beauty and their gentle nature. Yet we also understand that living amongst wildlife can be messy and frustrating and even scary. Some may even believe wild animals don't belong in cities at all. Our discussion tonight isn't about biological overpopulation causing suffering or starvation, or public health or safety concerns. Those aren't our issues. But really, how do we best protect nature and life within, particularly in the face of climate change and mass extinctions, pollution, and the devastating loss of habitat and wildlife worldwide? Animals share our homes and our planet. They are our family and our neighbors. They make us happy and they help sustain life on Earth. But as the human population grows and development expands, we find ourselves in closer quarters and in more conflict with wildlife, facing critical questions of what we should do with animals, perhaps because of their species or their numbers, in places some just don't want them. Whether it's deer or coyote, geese or swan, or even our feline friends, how do we best understand their role and impact on our local ecosystems, our own role and effect on those systems, and how do we protect nature given all the threats that we know now exist today? Well, as they say, knowledge is power. So tonight we'll be hearing about some important research we may have not heard before or considered, the role of ethics in conservation, and how we might all, human and non-human animals, find a place of sustainability, respect, and peaceful coexistence. So with that, I will turn it over to the great Dr. Oz Schmitz.
Thank you very much, Tanya. I have very fond memories of Ann Arbor when I was a doctoral student there. So it's nice to visit, even though visiting through a crazy medium right now is the only way I could be there. But anyway, I'm going to provide a brief presentation, a PowerPoint presentation. I don't want to drone on like a lecture, but I just want to present some ideas so that people can understand where I'm coming from in my thinking and why I approach, the whole issue of deer and deer management the way I do. So with that, I'm going to now launch the PowerPoint. I believe everybody can see that. Basically, the issue that typically I get called in to deal with is a question of should we cull or should we not cull deer? And it's usually pitched as sort of an idea of deer versus nature. That is, deer are having these huge negative, I drew the red arrow with a big minus sign. Deer are having a negative effect on nature or biodiversity. And there's a clash of values. People worry about the health of the environment, the quality of nature, the abundance of different species, biodiversity, and there's this perception that deer are quite damaging to nature or biodiversity. The problem is, a lot of times I find that nature or biodiversity is somewhat of a black box, and I'll get into that in a little later in the presentation when I try to unpack what I think is inside the black box. But to begin with, this whole idea of deer versus nature really results in the kind of thinking where as we increase deer abundance, or as deer abundance increases, we will see a decline in vegetation abundance because of what they eat, or loss of biodiversity as we increase deer abundance, because there are a lot of other species that rely on the vegetation for their habitat. So as deer increase abundance, they destroy the environment, and therefore we should get rid of them or reduce their populations so that we can bring things back up to higher levels of vegetation or biodiversity. And the way people often really look at the impacts of deer is through browse surveys, right? They want to provide evidence, which you need to make informed decisions. They bring in data from browse surveys where they go in the field and look at how much vegetation is eaten by deer. And this is a typical example of a heavily browsed stem. And then there's the lightly browsed stem here where you don't see a lot of bushiness. This is what we call coppicing. And so what happens is when things are heavily browsed, the plants compensate by growing more twigs, and then the deer eat more, and then it becomes a really bushy twig as opposed to this thin twig. And oftentimes, you see heavy browsing.
As soon as you get up, she's gonna be like,
Is it the same?
The problem the problem with these kinds of measurements, though, is you have to rely on the vegetation that's available in the field, in the stand or in a site. And that can sometimes vary in palatability. So deer may have different impacts, not because of differences in abundance of vegetation, but maybe some are more palatable than others. And so they have different preferences and forage differently. And so to get around that, there's another technique now. It's called Sentinel Plants. And what people do is they grow seedlings, tree seedlings, and take them and plant them in different sites. And they either plant them in the open or surround them with cages to exclude deer. And then what they do is they compare the browse impact between the two. And this is a very useful and highly appropriate scientific technique to really gauge the impact and not have it, what we say scientifically, confounded by the site conditions, because you're presenting the same kind of food in all different kinds of habitats. And it's a good indicator of browsing impact. Now, the thing we have to keep in mind is this is a browse study. It's not a regeneration study, though. So this is a way of just measuring what deer are doing in terms of their impact. And you can also then compare that to and identify what other species might be eating the seedlings and determine how much deer are having an impact versus other species. And so what you often expect is that in those exclosures that I showed you, the cages around the trees that prevent deer from eating, you would expect high plant height or abundance. And then
no labels on either
outside of
axis.
these exclosures
Like,
where
what-
deer are
what is
present,
it,
you
a
should
million?
see
A
less
billion?
vegetation.
One?
It makes intuitive sense. It's, you know, think about a candy jar or a cookie jar. If the jar stays closed and you can't get in, all the cookies are in.
I
If
work
you open
with.
the cookie jar and start eating the cookies, there are
I
fewer
know.
cookies.
We
That's
just have a
sort
whole
of the
data
idea.
science team, and they would love this graph.
And this was exactly the kind of study that Dr. Jacqueline Courteau did in this area in Ann Arbor, a Sentinel study to really measure the impact. And certainly, yes, she discovered
Just a
that
really
deer,
funny graph.
when they're present or have access to the
How many
vegetation,
steps?
they can have a pretty important impact on that. The limitation of that kind of study, though, is certainly you know what the vegetation should be when there are no deer, but when you have deer present, you don't know where along this axis you are here. And so you don't know if this is just a few deer or if it's many deer doing this damage. And so it becomes really hard to justify a cull based on this because you only have 2 data points. What you really need is to see if there is this linear trend between deer abundance. So what you have to do is this kind of study across a widely varying abundance of deer on the landscape in order to be able to argue that, yes, lowering deer abundance will actually end up improving the environment. So my experience, I actually did this with a master's student of mine back in 2008 and 2009. The article was published in 2010. And basically what we did was really go on a landscape level and look at the browsing impact and the regeneration impact across that landscape, ranging from low to high deer abundance. And I'll show you in a minute what that really means in terms of numbers. But we did it over the western Connecticut landscape, where this is different land cover types, where green is forest, red is more very urban environment, and the yellow in there is ex-urban environment. And you can see here, there's a widely varying deer density here, from hot spots to cold spots, if you will, or very low densities. And so what we did was sample plots in towns within each of these zones here. These are actually counties within the state, and we picked certain towns within each of these, and then measured vegetation in 10 large plots within each of those town areas. And so the density that we're talking about is a maximum of 30, where it's really dense, to a low of 5 deer per square mile in the northern part of the state. And so what we looked at was the stem density in 400 meter square plots. And we looked at both woody seedlings, so this would be looking at the regeneration stage, and we also looked at cover of trees in the plots. And what you find actually is that, yes, indeed, there's a lot of varying damage here, right? So sometimes you have 93 seedlings per plot, and then sometimes you only have as few as 46.1. It's hugely variable at the seedling stage, but it's not highly variable at the tree stage. So in other words, deer impacts here potentially do not affect the tree abundance here. There's regeneration happening even though there's variable amounts of woody seedlings present. And the reason why that's happening is just a basic ecological principle of self-thinning. So what happens is, especially when you're using hardwood trees like red oaks and things like that, or working in those kinds of forests, as you know, they've massed. There are seasonal boluses of acorns that hit the ground, and those acorns exceed the number that could possibly establish and grow in that landscape. And so a lot of them died before they even become seedlings, but there's still even a lot of seedlings that actually become established. But those seedlings, even if there was no herbivory, those seedlings would die because of competition. And it's the forest that's self-thinning that ends up reaching and creating this little variability at the tree stage. And so, again, what's happening here is we can see deer damage, but deer may not necessarily preclude the forest from regenerating. The other issue is the browse impact is a snapshot. It's only one side of the story. As I said, it's a browsing survey, it's not a regeneration survey. And so we can ask ourselves, what about regrowth or the productivity of that forest? And what you have to do then, and one useful way of doing that is what's called a coverboard technique, where basically if you look at these two kinds of stands right here, this one obviously has no understory. There are no seedlings growing up into the canopy, whereas you can see here in the left figure here, there's a lot of different height, vegetation at different heights in this canopy. And so with a coverboard technique, what you do is you put the coverboard in behind the vegetation and you stand and look at the coverboard and estimate how much of the coverboard at different heights is obscured by the vegetation. And so if a lot of it is covered all the way up the entire profile, then you will have what's called a high foliage height diversity index. And if there is no coverage, because of the openness, then you would have a low foliage height diversity index. And this is an indicator of how much forest regeneration is happening. If it's high, you've got good regeneration. If it's low, you wouldn't have any regeneration. And so what you might expect is that in areas where you have low deer abundances, you have very high foliage height diversity versus low foliage height diversity where deer are highly abundant. So we sampled across these sites, 10 plots in each of the towns we looked at. And again, I haven't drawn lines here because there's no relationship between the index of deer density and foliage height diversity. And this is just a summary across all towns because densities differed across the towns here also in terms of the mean and variance. So there is no negative relationship. Deer density is not a good indicator of the damage that you're going to see or the lack of regeneration or whatever you measure, right? So, the arguments then are always, okay, maybe they aren't impacting regeneration, but maybe they're affecting forest biodiversity or causing loss of native plants to invasive plants.
Look at this one.
And so you can also break things down. You can look at the vegetative cover, the species richness, that is the diversity by species identity of the different plant species in the forest, or the species evenness, that is how equally different species are represented, right? You could, it accounts for the dominance of different species. The percentage of ornamentals, these are the invasives that escape from people's gardens potentially. The number of native plants or the percentage of native plants in these areas. And basically what this value here, this R squared value indicates is how reliable, how important deer are in terms of explaining variation in each of these factors. A number close to 1 would mean deer are really, 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 really important. The fact that you get some numbers that are really tiny, and the fact that all of the numbers are less than 0.5, if you will, means that deer are having very little or no effect, even on the biodiversity across the Connecticut landscape, based on the surveys we've done. So the other issue that comes up, so in Connecticut, you could argue that a cull would not necessarily change any of the variables that I talked about, right? But even if people call for a cull, what's often frustrating for me when I get called in to give advice is people aren't clear about what goals they have. What are the measurable outcomes, right? It's typically deer are damaging biodiversity. Okay, so if we cull the deer, we'll get more biodiversity, but how much biodiversity is enough? And what do you mean by biodiversity? How much more do you want in terms of a goal? And because these goals are nebulous, you never really know how much culling you need to do to achieve any objective. And so, what we need to do is really clarify what our objectives are. One could be that we just want maximum forest cover where you don't have any deer. Or you might want deer at carrying capacity. And when you're dealing with hunters, they like to have a lot of deer, so you want deer close to carrying capacity, or maybe you want something in between also. And so it's really, really crucial to clarify what your measurable outcomes are. Otherwise, you will never know whether you're achieving them, and you will waste taxpayers' money in doing deer calls because you'll never know whether you've achieved your objectives or not. Now, if we're wanting something in between, like maximizing biodiversity or whatever, then here's where we have to really look inside the black box and think about the ecology of the situation. And so,
that's a good one too.
For me, I don't think of deer versus nature. I actually think deer are part of nature. They are biodiversity. They're part of biodiversity.
That sounds okay.
And what inspires me as an ecologist is really to look at this complexity head on and try and understand the complexity and really look into the black box and try and understand what's going on. And one of the things we know is that it's dynamic. That black box is dynamic. Where you are in the black box depends on the habitat structure.